Protein synthesis is the process by which your cell takes DNA that's found in the nucleus and transforms it into RNA, which leaves the nucleus and goes into the cytoplasm of the cell. And then the RNA is used as a template to make a protein. So that is the flow of genetic information in the cell. You start with DNA, which is found in the nucleus of the cell. It's made up of chromosomes, and those chromosomes are composed of genes. Each of the different genes will code for specific proteins. So the DNA gets transformed into RNA. The RNA leaves the cell, goes out into the cytoplasm, and basically gives directions for the protein to be created, to be made, to be assembled. If you were going to use an analogy, what you could say is that DNA is the recipe book for all of the different proteins that your cells could make. Every single one is found in the DNA molecule. And then RNA would be, if you want to make just one of those recipes, you might write down that specific recipe on an index card or on a sheet of paper and take that into the kitchen with you so that you don't make a mess on your recipe book. But that particular index card or piece of paper would be similar to the RNA molecule, because you're going to take that kind of out into the actual process to make the protein. And then the protein would be analogous to your final product. So if it's a recipe for a chocolate cake, then the chocolate cake would be similar to the protein. DNA and RNA are similar in that they're both composed of nucleotides, but they're different because DNA has a different sugar. So the sugar for the DNA molecule is deoxyribose. And then for RNA, the sugar is ribose. One of the other ways in which they're different is that DNA is double-stranded, whereas RNA is single-stranded. And so when you see a double helix, that's always a DNA molecule, whereas RNA is just a single strand. The other difference is kind of interesting, and that has to do with the specific bases that the molecule has. So DNA has the 4 bases we've talked about before. So it has adenine, thymine, cytosine, and guanine. And according to base pairing rules, whenever you have a DNA molecule, the adenine is always bound to a thymine molecule and vice versa. And then the cytosine is always bound to a guanine molecule and vice versa. RNA is a little different. It has the adenine and it has the cytosine, it has the guanine, but instead of thymine, it has a molecule called uracil, which is indicated by a U. And so when you talk about base pairing rules according to RNA, instead of the A binding with the T, the A actually binds with U, with uracil, and then the C and the G still hang out together. So this is important because part of this process of going from DNA to RNA is you look at a DNA strand and you make a complementary RNA strand according to base pairing rules. But since RNA doesn't have thymine, you sub out the uracil for the thymine that would be the complementary strand of DNA. So let's I'll give you an example of that. And this actually is kind of the first step. This is step 1, transcription. So if you have a DNA strand, and that DNA strand, let's say it has, the part that we're interested in has 6 bases. So let's say you got A, T, T, C, G, A. Okay, so this is the strand of DNA that we're going to make a complementary RNA strand to. So the RNA strand that we made that's complementary to this, according to base pairing rules, according to RNA base pairing rules, the adenine is going to bind with uracil, the thymine would bind with adenine, the second thymine would also bind with adenine, the cytosine would bind with guanine, the guanine would bind with cytosine, and then the adenine would bind with uracil. So the original DNA strand reads ATTCGA. The complementary RNA strand reads UAAGCU. Okay. Now the way this happens is you have an enzyme called RNA polymerase, and you can see it in the picture to the left. It's a very, very big molecule. It will bind to a region of DNA, and it binds just behind a specific sequence of DNA called a promoter. And that promoter tells RNA polymerase where to bind and where to start transcribing the gene. So RNA binds to the DNA strand, unwinds it, and then assembles a messenger RNA transcript complementary to the DNA strand that it cares about. And so that's step one, that's transcription. This takes place in the nucleus. And then the mRNA transcript is going to leave the nucleus and go out into the cytoplasm of the cell and get involved in kind of the next step, step two, which is translation. All right, so let's go down here to translation. So translation takes place in the cytoplasm, and you need, for this process, you obviously need your messenger RNA strand. You also need ribosomes. Ribosomes are an organelle in the cell, and they are composed of protein subunits, a large and a small one, and also a particular type of RNA called ribosomal RNA. And so that ribosomal RNA helps make up the ribosome itself. You also need a supply of amino acids just kind of floating around in the cell, and you need something to carry the amino acids to the ribosome and to the messenger RNA. So the material that does that, the material that carries the amino acids to the ribosome, that is called transfer RNA, or tRNA. So it's transferring amino acids that are floating around in the cytoplasm. It's transferring them to this new protein that's being created, this new polypeptide chain that is slowly growing as each amino acid is kind of added to it. The other thing you need for translation to take place is energy. So that energy is going to be in the form of ATP, because that's the energy currency of the cell. So translation kind of is broken down into three separate things that occur. So the first thing that's going to happen is the ribosome is going to assemble around the messenger RNA strand. And so in the picture, what you can see is that the large subunit of the ribosome and the smaller subunit of the ribosome have kind of sandwiched this messenger RNA strand between them. So the ribosome assembles around the messenger RNA. And when it does that, it's going to expose basically 3 nucleotides of the messenger RNA at a time. And those 3 nucleotides are what we call a codon. And you can think of a codon like a special code. So the 3 nucleotides of the messenger RNA that are exposed at a time are called the codon. These codons are going to coordinate or match with an anticodon that's found on transfer RNA. So when we talked about up here our DNA strand and then our RNA strand, if we're talking about a codon, it's going to be 3 nucleotides. So the first codon of this RNA strand would be UAA. So if our RNA codon, messenger RNA codon, is UAA, then the anticodon is going to be complementary to that according to base pairing rules. Remember that uracil is a substitution for thymine, and according to base pairing rules, thymine binds with adenine. So the first nucleotide of the anticodon would be adenine, and then A binds with T, and then the second A would bind with T. So if you got a gene on the DNA that is ATT, the messenger RNA strand that's complementary to that would be UAA, and then the anticodon that's found on the transfer RNA would be complementary to the codon on the messenger RNA, so it would be ATT. So notice that the anticodon in most cases is very, very similar to the original DNA strand. Now, let's look at this one. So let's say the second codon here would be CGA. So if the codon was CGA, the anticodon would then be, well, C binds with G, G binds with C, A binds with T, but remember we don't have thymine in RNA, we've got uracil. So the anticodon would be GCU. If the original DNA strand was CGA, the transfer RNA anticodon would be GCU. All right, so this is why base pairing rules are so important, is because they're used throughout this process of protein synthesis. Because every single time that they're binding according to the base pairing rules, where A binds with T or uracil instead of thymine, if you've got that on RNA, and then C binds with G. All right, so that was kind of the first part of translation, is the ribosome assembles around the messenger RNA, exposing the codon. The second step has to do with these codons and the transfer RNA. So the second step is that the transfer RNA molecule is going to bring an amino acid to the ribosome and the messenger RNA. And it does that, as we said, according to the codons and the anticodons. So on this piece of transfer RNA, you've got a region that is the anticodon that's going to bind to the messenger RNA, and then you've got a region up top where an amino acid is bound, okay? And each transfer RNA, anticodon, and codon, each of these pairings is very specific to one amino acid. So if there is a codon on the messenger RNA, that is going to code for one amino acid, and that's important. So each codon codes for one amino acid. So you're not going to have a codon on the messenger RNA that's ambiguous, where it could be one of several amino acids. No, it's going to be specific. It's going to be one amino acid. Now, there is some redundancy in the genetic code because there might be more than one codon per amino acid. So, for example, let's just say that the GGG here, that could code for alanine, but then the UUU might also code for alanine. So each codon is going to be specific to an amino acid, but there could be more than one codon per amino acid. And that's actually very important because it helps protect against mutations causing problems with protein synthesis, which we'll talk about in just a minute. But so each codon codes for one amino acid, but there can be more than one codon per amino acid. So that would be the second step in translation. The third part of translation is basically after all this has happened, after all of the strand of messenger RNA, it's gone down, they've added amino acids, this protein has grown from all of the amino acids being assembled. Once that occurs, the protein is actually going to, well, we'll call it an amino acid chain, so a polypeptide chain. It's going to actually assume its 3D shape to become a protein, a fully activated protein. So it's going to disconnect from the ribosomal subunit and then kind of go out, and there are other proteins called chaperone proteins that will actually help it fold into its final 3D shape. And then all of these components just kind of disassociate. They just kind of break apart. They go off and go about their business until the next time they need to make a protein. So that is how protein synthesis occurs in the cell. The next thing we're going to talk about in the next video will be kind of a quick overview of mutations and genetically modified organisms.